we are looking at membrane structure and function. If you've not already done so, make sure to get the warm-up done. There are two essential questions for the day. Where are membranes found? And then we want to relate the structure and function of membranes. So we need to look first at what they physically are made out of and what they look like, and then how that relates to what they actually do. To understand membranes, we need to spend some time going back over units 1 and 2. So here's a quick review. We're looking on page 81 of our notes about cells and chemistry that we need to do to understand cell membranes. So for starters, what are cells? And the two basic types are prokaryotic and eukaryotic. Remember, pro, no, euc, nuke. So eukaryotes have a nucleus and they have membrane-bound organelles. The membranes are also going to be found around all cells, regardless of whether they're prokaryotic or eukaryotic. Even if they have a cell wall, they're going to have a cell membrane. So all cells have to have cell membranes. And we call this membrane that goes around the outside of cells either the cell membrane or the plasma membrane. Okay, so here's our diagram of a typical cell. And when we're talking about those membrane-bound organelles, what we're really talking about are anywhere you see these like sheets of material here around the whole thing, around the nucleus, and our simplified diagram here is going to highlight some of those big ones. Again, this is only going to be true for eukaryotic cells, your plants, animals, fungi, and some other organisms that we collectively call protists. So we've got the nucleus, obviously. Right outside of the nucleus is another layer of membranes that are all folded called the endoplasmic reticulum. Remember that those can be smooth or rough. Rough would have ribosomes on them. The ER is going to be responsible for taking the proteins that ribosomes make and kind of preparing them and helping assemble them, and then shipping them off to these little Amazon warehouses out here. Those would be the Golgi apparatus or the Golgi complex or Golgi bodies, all the same thing. And the little Amazon packages that are made by the Golgi, these little guys here that are full of different chemicals, are going to be your vesicles. We also looked at the fact that all eukaryotic cells have mitochondria. That's why all eukaryotic cells are able to do cellular respiration. And now, really importantly, we need to look at how the parts of the membrane interact with water, because that relationship with water is going to determine how the membrane assembles. So we need to review chemistry. Remember that when we talk about covalent bonds, we're thinking about chemical bonds where electrons are shared. So covalent bonds can be either polar or nonpolar. When we're talking about a polar covalent bond, we're thinking about the electrons being shared unequally. We see this with oxygen and nitrogen especially. Oxygen and nitrogen are both very electronegative. They're going to pull the electrons towards them, while carbon and hydrogen are not as electronegative. So those will make the very unequal sharing of electrons and the polar bonds. When we talk about molecules that have polar bonds, these are going to collectively be called hydrophilic. Hydro for water, philic meaning loving. So water-loving molecules that do mix with water. And your prime example there would be, of course, water, as well as salts and other hydrophilic substances. Anything that does dissolve in water is hydrophilic. On the nonpolar side, we're looking at things where the electrons are not shared, that they are shared equally. And that means that there's not going to be any kind of charge whatsoever. The electrons that are negatively charged are going to be spending their time equally between both atoms involved in the bond. So carbon to carbon or carbon to hydrogen, where they're sharing well, and that's going to make them hydrophobic. They do not mix well with water. Make sure to circle that do not there. Hydrophobic meaning water fearing. And your examples are fats and oils, your lipids. Remember, we talked about how in hydrophilic substances, in things that are polar, we're going to have hydrogen bonds that form between the molecules. So here is one molecule, so an atom, an atom, an atom, they're bonded together. That makes a molecule. In this case, we're looking at water, and different water molecules can interact, making a hydrogen bond. So anytime we have anything polar, they can make hydrogen bonds. Okay, We'll also spend some time talking about the different components of the cell membrane. So we need to know what the molecules are that make them up. These are your different macromolecules, remember, your carbohydrates, lipids, proteins, and nucleic acids. When we look at the elements for carbs, we're thinking carbon to hydrogen to oxygen in that 1 to 2 to 1 ratio. And because of how much oxygen are in these carbohydrates, they are polar. So these are going to be your sugars, and make sure you also include that your monomer here is a monosaccharide, your prime example being glucose. So carbohydrates or sugars are going to be found in cell membranes a little bit. Lipids, recall, are mostly going to be made up of carbon and hydrogen. Because there's no oxygen or nitrogen or very, very little of it, these are going to be nonpolar, so they do not mix with water, and we're thinking our fats and our oils. I don't have it written up here, but you should also include that this top part here makes up what is the glycerol portion of a lipid, and then your fatty acids are the long chains of carbon and hydrogens that come down from that. Next, proteins. They have the monomer amino acids, and those are going to be made out of carbon, hydrogen, oxygen, nitrogen, and sulfur. These can either be polar or nonpolar, and your examples are really plentiful. Remember that proteins can have tons and tons of complex three-dimensional shapes, and that's why they are so versatile. Now, for nucleic acids, they're important, but we're not going to see them in this section. They don't have as much to do with the membrane, so you do have that filled in your chart in the notes, but we don't need to go over that. So now let's look at the membrane structure. And in fact, if we take a look at pages 82 and 83, this is all about membranes. The structure is going to be on page 82, and then the function and how different materials move is going to be on 83. So that function is going to be based on the structure. Okay. So there's really a few different things going on with membranes. The biggest component of cell membranes are going to be what are known as a phospholipid bilayer. And as you can tell, a phospholipid bilayer is a phospholipid in 2 layers. So the phospholipid is a molecule that's really, really similar to a fatty acid, to a lipid, which has some fatty acids and then the glycerol. But now instead of having just a glycerol with another fatty acid, we are going to take a phosphate group. And that has a lot of oxygen and phosphorus in it. So that is going to be really, really hydrophilic, very polar. It interacts well with water. The lipid tails, on the other hand, well, those are going to be hydrophobic. They do not interact with water. So we have a molecule that has a part of it that interacts with water and a part of it that doesn't. So as I said, the bilayer means that we have 2 layers, and those are made up of the heads and the tails oriented in such a way that the hydrophilic heads face outward, which would be towards the water, and the hydrophobic tails in the middle kind of get tucked in. When we look at how membranes interact with water, so the inside of the cell and the outside of the cell are going to be water-based, aqueous. So that means that all of these little hydrophilic heads will be fine with interacting with water. Same out here, but the little hydrophobic tails, the lipids, the fats, get tucked inside. So that is the main component of the cell membrane, is the phospholipid bilayer, but there's also other stuff embedded in it. And so for that reason, the model of the cell membrane is also often called a fluid mosaic model. So just like a city with a whole bunch of people walking through it, the fluid mosaic model describes the cell membrane where all of these individual phospholipids can move around each other, and we can have stuff embedded in it. So on your notes, you're going to notice that you've got a whole bunch of bullets there, 3 bullets, for what can be embedded in the membrane. So we start with proteins. The majority of membranes are made out of the phospholipid, and then proteins are in second place. There's a lot of proteins in our membranes, and those proteins can go all the way through like we see here. We can call that an integral protein, or it can be embedded just on one side, either the outside or the inside, and we would call that a peripheral protein. And just like proteins everywhere else in the cell, they're very, very diverse and they have a lot of functions. So they're going to be important for transporting materials in and out of the cell, acting as enzymes, communicating with different cells. The list goes on, but those are going to be the main ones. Another component of the membrane that we see are going to be these little 4 ringed structures here. These are called cholesterol, and these are lipids that get integrated into the hydrophobic center. So cholesterol is hydrophobic because it is a lipid, and it interacts well with the other hydrophobic core part here. Especially in animal cells, that cholesterol is going to be important for helping this whole membrane stay fluid and flexible instead of kind of congealing and turning into lard, really. And then lastly, we're not going to distinguish between them, but we have what are called glycoproteins and glycolipid. Glyco meaning sugar. So this is either going to be a protein or a lipid bound to a carbohydrate that sticks usually out of the cell. And glycoproteins and glycolipids are really important for allowing the cell to recognize other cells and for communication. So, for example, when you get a bacterial infection or you get bacteria in your blood, it's going to be some of these glycoproteins and glycolipids on the bacteria that are recognized by our immune cells as not being human. Okay. If you need to review and rewind, you can go back and take a look at what all of the different parts are, but you should take a moment and use this key to color your membrane that is at the bottom of page 82. The hydrophobic and hydrophilic parts refer to the phospholipid bilayer. There's going to be 2 membrane proteins. You don't need to distinguish in your coloring which one's which, but there's an integral and a peripheral. Color in the 2 cholesterol molecules and the 1 glycoprotein or glycolipid. Okay, and now we can continue on to the next page and then we'll be done. So we want to look at how materials move, because what is the ultimate role of the membrane? Well, it's to act as a bouncer. So what membranes do are to help regulate the movement of materials into and out of the cell. Just like a bouncer is going to be at a club watching who comes in and who leaves and making sure that everything's running smoothly, that's what our membranes are doing, is letting material from the environment get into the cell. When substances can move through the membrane, then we say that it is permeable to the substance. So oxygen can get through the membranes very, very easily. We say membranes are permeable to oxygen. And because they're not permeable to all substances, we're going to say that membranes are semi or selectively permeable. Some things can go through, others can't. Okay, and so let's pull this back to what we've been talking about, which is homeostasis. It isn't enough to get material, whether that's glucose or oxygen or other nutrients. It's not enough just to get it to the cell. The material has to get into the cell where it can be used. So for example, oxygen needs to go from the lungs, diffuse across membranes to get out of the lungs and into the blood, continue moving in the blood, and then diffuse through a couple of membranes through the blood and into the cell where it's going to be used. So for us to be able to maintain homeostasis, our cells need to be able to get what they need. So the blood is going to carry the nutrients to the cell, carry waste products away, and then the membrane is where materials are actually passing through. So to understand how things move, let's take a minute just to talk about how particles move. And I'm going to use like molecule and particle kind of interchangeably here, but remember we're thinking tiny, 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 tiny. So atoms make up the molecules that we're talking about here. A whole bunch of molecules can come together to make a macromolecule. Organelles are way, way bigger. So these are tiny little components of a cell, okay? And so each cell is going to follow the same general trend, each particle is going to follow the same general trend, which is that all the substances will move completely at random. So if you watch any given particle here, you'll see that they just kind of bounce and their direction is totally random, okay? But as a whole, we say there is a net movement of particles from areas of high concentration to areas of lower concentration. For example, if you were mixing Kool-Aid into water, you're going to start with one area that has a lot of Kool-Aid, but as you mix it and let it set, eventually it's going to spread out. So we refer to this as a concentration gradient, where you have one area that has more and one area that has less. So a greater concentration on the left side of the diagram and a lower concentration on the right. And materials are going to have a tendency to move until each of the particles is in equilibrium, meaning that even though they're all still moving, overall they're pretty well dispersed. I kind of like to think of this as if I was to take a whole class of like kindergartners the day after Halloween when they're all hopped up on candy and bring them into a gym. And at first, they're all kind of huddled near the door. But as you say, hey, go play, they're going to disperse. And eventually, they're going to fill up the whole gym. And at any given moment, little Timmy or Tommy or Susie might be anywhere. They could be bouncing off of each other over on one side or the other. But overall, they're pretty well distributed. And we call that equilibrium. So particles tend to move towards equilibrium. So let's wrap up this page and talk about the general trend and the general way material can move through a membrane. Okay? 3 ways that materials can move into and out of cells. And what's important to remember here is it's about the concentration gradient, not about whether the particle is moving in or out. So we start with what is called passive transport. And this is when materials are going to move across the membrane down the concentration gradient, which means going from an area of high to an area of lower concentration. So if you're going in the direction that particles tend to move anyway, which is down their concentration gradient, right? You start with this gradient and the general movement is that it's going from high to low. Well, that would be our passive transport. The second type that we'll look at in this unit is called active transport. And as you can imagine, active transport is going to need to use energy. And what this is going to do is go against the concentration gradient. Think about all those kids that were running around like crazy in the gymnasium. When it's time to round them up again and get them all in one area, so instead of being at equilibrium, there's one area that has a lot of kids, you're going to need to put in some energy to wrangle them all up. And active transport is going to do the same thing. It's going to push material from one side to another against the concentration gradient, meaning that they'll move things from an area of lower to areas of higher concentration. And then finally, the last type of transport that we're going to look at is called bulk transport. And this will also be a type of active transport in that it is using cellular energy, but instead of individual particles moving across the membrane, we're going to take big chunks of material, take it into the cell, but we will look at that further. Okay, so with that, there was a lot of information gone over. Review it, make sure you're highlighting your notes, make sure you're finding those page numbers, and then you can get to work on the crossword puzzle. Let me know if you have any questions.